Good afternoon, everyone. Welcome to Wake Forest Center for Artificial Intelligence Research, Artificial Intelligence in Action seminar series. I am Metin Gürkan. I'm the director of the center, and it's a pleasure to introduce today's distinguished speaker, Dr. Gustavo Rohde. Dr. Rohde is an associate professor of biomedical engineering and electrical and computer engineering at the University of Virginia. And we just checked out, it's 3 hours, 20 minutes from here. And he holds his undergraduate degree in physics and mathematics and master's degree in electrical engineering from Vanderbilt University. And he earned his PhD in applied mathematics and scientific computation from the University of Maryland. His research focuses on computational modeling and machine learning techniques for biomedical imaging analysis with applications in disease prognosis, the diagnosis, prognosis, and understanding biological and physiological systems. I have known Gustavo for many years, and I've admired his work. And just, he's a phenomenal scientist, but at the same time, he's such a humble and just approachable person. I always admired his personality as well as his scientific output. Today, Dr. Rohde will present on transport-based morphometry for predictive modeling of biomedical imaging data. While AI and machine learning are widely used for predictive modeling in biomedical imaging, they often face challenges such as lack of interpretability, robustness, and high computational costs. In this talk, Dr. Rohde will introduce an alternative mathematical modeling technique based on mass transport theory, an approach that underlies molecular and tissue distribution in biological systems. And he will demonstrate how this method can mine information from digital microscopy images in cancer research and MRI scans in autism studies, ultimately leading to more interpretable and accurate predictive models that provide deeper insights into underlying physiological processes. This is quite a spectrum. Just it's really, really wonderful. Following his presentation, we will have time for questions. Please feel free to submit your questions in the chat, and we will address as many as possible during the discussion. With that, I am honored to turn the floor over to Dr. Rohde.
Thank you very much, Metin. Thank you very much for the very kind words. I appreciate everything you said. And yeah, I always enjoyed interacting with you. Hopefully we can get together very soon. We can bring you here for a seminar in person, share up here as well. So the first slide, I think I'm supposed to let you guys know that there's some credit that if you follow these directions here, you can get. If you watch this slide, if you watch this presentation, follow this procedure here, use the Northwest Area Health Education Center SMS text message for attendance. If you have any questions, contact this email. You can see my mouse when I wiggle this, right?
Yes.
Okay, yeah.
Yes.
So that's the email to contact for help, but there is credit that you can get for watching this presentation, at least something, right? So so we can go ahead. This is the title. And so my contact information is here, Gustavo Rohde at Gmail, or just Gustavo at Virginia. I'm a professor at the biomedical engineering and electrical engineering departments here. There's a little bit, our website is imagedatascience.com. And today I'll be talking about our transport-related projects. So if you just go imagedatascience/transport, you'll see many of the papers and so on here as well. First and foremost, right, give credit and acknowledgement that we all of the stuff that I've done here, it was mostly done by many other people. And right now, personnel in our lab include Christopher Pass, Nakib Hassan, Hongyu Khan, and Natasha Aronside, and Ivan Medri. They're doing a lot of the calculations I showed, but there are many people in the past that also have contributed, as well as mentors and as well as NIH and ONR funding. So let's begin, right? So controversy, right? Quick question: what is biomedical data science? In this case, with imaging, right? In my view, it's related to it's an activity related to a type of question, right? So, for example, here somebody shows up to your office and say, "Hey, I have these brain scans, these brain MRIs one type of population, right? And in this case here, they're persons with low fitness index, is a class 1. And I have these MRIs of class 2 with a person with high fitness index, right? They will come to you and may ask a question: is there a, is fitness and gray matter morphology related somehow? Is there a difference between these two things, right? Or if you're working in pathology or cell biology or something like that, you may measure cells or subcellular components. In this case, I'm showing images of nuclei extracted from digital pathology images. But the same question, like say category 1, you extracted a bunch of nuclei from a benign version of a cancer, and extracted nuclei from a malignant version of a cancer. Typically this type of biomedical data science involves two questions. Question number 1 is that you should ask, is there a difference in what you're measuring, right? In this case, for example, is there a difference between in the gray matter morphology between persons with low fitness index and persons with high fitness index? If you just look by eye, very difficult to tell, right? But also being humble, understanding the position of a scientist, which is a position of, at the beginning, being ignorant, right? So you would, a person here will say, well, actually, these are three-dimensional images. You're observing 1 million of voxels or so. Very difficult for your brain to examine all these million voxels and come up with a definitive answers. Yes, there's a difference, or no, there isn't a difference, right? So when data is so overwhelming like this, it could be very rich, and it could contain answers. It could also not contain answers. So this is the questions that biomedical data science try to answer. And if you do find some statistical evidence, for example, saying that there is a difference between these classes, benign cells are different from malignant cells, for example, then the next natural question to ask is, what is the difference, right, as far as that you're observing? So that's the topic that we're working on, right? And today, we're not going to work sort of blindly here, right? We're not going to solve all problems of this nature. Rather, we're going to solve a specific, we're going to present a solution to a specific category of subproblems in this area that have to do with modeling mass distributions inside cells, tissues, and organs, right? So that's really, and so let's start at the beginning, more or less like a biophysicist would do, or a physicist would do, right? Let's look at the movie of a cell moving, right? Doing its thing. In fact, you're not looking at the whole cell, you're looking at only a protein of the cell, right? It's actin protein, which is a protein that helps form actin filaments, which are the cytoskeleton of the cell, gives the cell some mechanical properties, is related to the movement of the cell, and so on. So you're imaging that particular protein, right? Amongst thousands of choice, you're imaging a particular one. Or, here is a brain of a child growing from 0 to 48 months, right? Taken every month or every 2 months or something like that. So there is a dynamical process happening here. There's a dynamical system, right? And it's a mass distribution dynamical system, right? It's because you're imaging mass here. In here, you're imaging how much protein that is, right? The brighter the pixel, the more protein, the more mass. Here as well, this is a proton density weighted image, which proton density, right? The higher the intensity, the more proton density there is. So it's a mass distribution image. And if you ask a physicist, say, hey, can you come up with an equation to describe this dynamical system? The natural starting point for anybody doing this kind of thought process would be the following equation. Let's say that the image here, or the intensity, is proportional to mass, right? So you would ask, what is the differential equation? What is the di dt, right? First of all, the mass distribution is a function of x, y, z, and time. That's our dynamical system. That's our mass distribution. And if you say, okay, let me fix a coordinate here, right? This voxel, this red voxel that I'm putting here, or this one, doesn't matter. And I want to say, how does this intensity in this voxel, this pixel, change over time? That's a di dt. So a physicist would put two terms in the other hand side of the equation. The first term is related to, if you fix on a voxel, there is mass coming in and there is mass coming out. So the mass coming out minus the mass coming in is what we call flux, right? And it's given by this term of this differential equation, which has to do with the velocity flux, right? So you multiply your intensity or your mass by the velocity and take the gradient, which is dx, dx and dy, the partial derivatives, and that gives you the total flux or the net flux of mass through this volume, through this voxel, right? And you also have to realize that, you know, in this case, the baby may be eating food, so there may be mass being deposited there, right? Or in this case of the cell, protein decomposing or mass being removed, right? So you will also have a source and a sink term. This is not a very controversial statement, right? This is where any biophysicist would start, a starting point. Now, of course, we don't know what the flow is, and we don't know what the source sink terms are, but primarily you do imaging to find out some of these terms and do the science to answer those questions that we just asked, right? One thing to note that in the majority of the imaging modalities at departments such as yours and ours, it's not yours, but anybody has it, these images are not calibrated. So unless you have some very calibrated experimentation with actual physical units, right, for actual kilograms per area, per volume here, you're not going to be able to calculate any source or sink term. So typically the analysis that we do and anybody does without calibrated has to neglect this part, right? But these other parts will serve as a starting point. So what is mass transport? A little bit more, right? So we can, let's consider a starting point and an ending point for a nucleus, right? So the brighter the pixel in here, the more chromatin is in that pixel, right? So, and let's imagine that there is a time transition that goes from this one to that one. How does this work? Well, we can imagine that there is a velocity, some force making particles move from the center and being pushed out, right? So this is our initial condition. This is the end condition where we'd like to have. And you can see little by little mass is coming from here, from the center, and being pushed out to the periphery. This is what we call transport, right? And in this case, we are assuming that the total mass here is conserved. Again, in imaging, typically we can't assume anything else because we don't have the actual calibration. But this is what transport is. This is what this velocity field does. And using this velocity field, we can relate the velocity fields to a displacement field, right? And if you want to write this image here as a function of time, so space and time, using the displacement field, this is it, right? So this is basically this change of variables that we have all seen. This is what we're calling transport or conservative transport that conserves mass, right? And it follows that this mass conservation is a direct consequence of the fact that we are dealing with uncalibrated imaging. So we don't have the source or sink term. We can't use it anyway. So we set it to 0, which gives us a mass conservation equation. And, you know, there's a special type of motion that is constant velocity motion where, you know, where these particles are not being pushed in a zigzag way, but they're going to be pushed in a straightforward, like, you know, the particle that started here moving that way keeps moving that way until it's deposited that way, right? That's a very special type of transport and is known as optimal transport, right? That minimizes the total mass times distance that requires to get from one configuration to the other configuration. If you try to minimize that, find the V that minimizes this whole thing such that the continuity equation is satisfied, such that the beginning point is 1 and the ending point is 1, then this F characterized by the Granger convex function solves this metric and is known as the optimal transport, right? So I'm not going to go much more deeply into what this means because I want to go into the transport-based morphometry and try to understand how we can use these concepts to extract information, right? So this is pretty much, I think there's one more slide about it, but not much more. But basically the gist is we're going to rearrange mass from one configuration to the other one in such a way that there's minimum effort. We're going to imagine that the cells, the brains, tissues, etc., work in an energy minimization, energy conservation principle. I want to find what's the least amount of energy a cell has to spend to reconfigure itself from this configuration to that one. That's known as optimal transport. There are theorems that show you characterize what this velocities push forward, this functions maps that should do, and those are the ones that we're calculating. A little bit, you know, I mentioned already change of variables. This is essentially what it is. Let's say we have a distribution here in 1D. We all have done this, I think, in calculus, basic calculus, right? Let's say we have a 1D distribution here. There's a mass distribution, integrates to 1. It's all positive and very good. And then let's say we have a map, G of X, right? If we compose our mass distribution with G of X and multiply by the derivative of G of X, we can find, you know, we can use whatever g of x we want to use to transform this guy into pretty much anything we want, right? And so here are some of the things, the games you can play. You can transform this one into a variety of different things through this equation. We can translate, right? We can make the distribution fatter or thinner by doing scaling. We can apply non-rigid deformations. We can wiggle this function any way we want, right? So the modeling that we're going to propose starts from, let's say, a reference point here. And now instead of modeling functions directly here, we're just going to say, okay, if I throw in different g's here, I can build any function that I want. And that's what we're going to call the basis of our transport-based morphometry framework, right? Of course, this can be done in one dimension, but in two dimensions as well, right? So we can take an image like this and turn it like this by choosing some kind of unique G, right? And again, this can be 1D translations, 2D translations, scaling, anisotropic scaling. You can rotate this image. You can redistribute mass in any way you want, right? This is basically the change of variable. When I put the D here, I mean the Jacobian of the deformation here. When I put G prime, it's the derivative of deformation because there's only one G dimension, right? So that's the mechanism. We're going to push mass around, intensity around using these equations, right? And we coupling that notion with the notion of optimal transport to find unique correspondences, right? Image minimization transport maps. In the past few years, our laboratory actually, I'm not going to go into much detail here. This will be one slide introduction to this. But this can constitute a new way to represent functions, represent densities, represent images, right? So you can think of this as the same way as you think of a Fourier transform. So we're going to use this concept to build a new kind of mathematical transform for signal and images, right? So the input space would be the space of images, which in this case, we're assuming they are probability density functions or mass distributions, right? You're imaging mass in the cell, you're imaging mass in the tissues. So this is your input space. And the transform space are going to be the diffeomorphisms, meaning the transport functions that map your data to a chosen reference, right? So we're going to choose a reference and map an image to the reference or push the mass from this image to the reference in such a way that the push is given optimally, is minimizing mass times distance, right? So it's an optimal transport. And using optimal transport theory, we can guarantee there's a unique F, right? So the point being that any image here can be associated with a unique diffeomorphism. Just one. Which one? The one that pushes the mass from the reference to that in an optimal way, optimal transport. Optimal transport theory gives us this unique reference, right? And so it has a forward transform, which are these deformation maps associated with this operation, and has an inverse transform, where you can then take this map and push the reference back into the image. So instead of dealing with these images here, now we're going to deal with these transportation maps, and that's going to be our transform. And we're going to use these transport maps to solve all the problems that we're going to solve: classification, regression, and so on. There's a series of papers that our lab has worked for the past 5-10 years building these mathematical representations, and today we're just going to use them to do medicine and biology. So starting with a little bit of intuition, right? So if I ask you a crazy question and say, okay, you can look at a bunch of images of nuclei, but what are all the possible ways that one nucleus, that chromatin can be arranged within this nuclear envelope? Really crazy question, right? But in fact, this mathematical equations give you an opportunity to model, to come up with them, right? So for example, you can say, well, chromatin can vary from being placed in the center towards the edge or vice versa, right? Chromatin can vary, making the envelope more elongated or less elongated. Chromatin can vary by having some bit of mass here being pushed there, and all the options in between, right? And any mixtures that you want between these as well. So we have now a powerful framework to model mass distributions inside cells, tissues, organs, etc. Right? Of course, I showed you 4. I can show you. How many can I show you? Infinitely many. Using this formula. All I need to do is come up with Fs. How many Fs are there? Infinitely many. So it's a very powerful modeling mechanism. I can model any image of a nucleus with starting from a reference and finding the optimal transport map that maps this reference to the nucleus. So I have a modeling technique. And so this is the summary of this representation. We can work with the native space of images, right, to try to do our machine learning, which a lot of people do. But as you know, it's very cumbersome. I think Metin cited some of the things I wrote in the abstract, right? You really don't know what functions you should be fitting for classifying this versus that. And you don't know if you come up with a function that seems to do well in your training data or a small set of test data, who knows what happens in the real world, right? You don't know the performance. Like, it's a black box kind of approach to science. Here we have a little difference. We have a nonlinear signal transformation or image transformation that take your data from PDF space to the diffeomorphisms, where we have some theory that some things can be linearized. So, and all the proposition that we're doing in this talk is, instead of doing machine learning in the image space, do it in this transform space. There are some mathematical properties that show we can convexify these classes. And the intuition is that, here you can look at the Euclidean geodesic between this image and that image, right? Or the transport geodesic. This approach here is going to push mass around, right? It's a very convenient mathematical formulation to push mass around, and this is going to form the basis of our model. So that's the introduction I wanted to give. So let's talk about transport-based morphometry. This is a new thing that, we've written papers 5-10 years about it, but it's a new terminology. It's a little bit stolen, maybe Metin works in the same field or used to, perhaps, people did registration-based work to analyze morphometry of brain images typically, right? They would take an atlas, register all the images to the atlas, and do deformation-based morphometry or voxel-based morphometry. We borrowed the nomenclature here, and we are now trying to introduce this concept of transport-based morphometry. It has a lot of advances. I'm not going to have a slide comparison, right? We can talk more if anybody wants it some way. So here's the transport-based morphometry in a nutshell, right? It's a very contained, very simple pipeline, where first you compute this transform that I mentioned, and we have codes to do a lot of them, right? I'll show you the web page from the GitHub page at the end, where you see where the codes are. But first you compute the transform. And what are the transforms again? Well, it's a unique way to associate an image to a deformation map, right? Which means you push this reference to that image in an optimal way, and this pushing function is this f is uniquely associated with that image. So whether you have this image or the f, it doesn't matter, because if somebody gives you an f, you can recover the image. If somebody gives you an image, they can recover the f. But our proposition is, once you get this transform or these transport maps, you're going to do all your regression to solve your biomedical problems on this transformation space, on this space function of f's. And I'm going to show you the advantages of doing that, right? What regressions people normally want to do? They want to understand variations. They want to do PCA, understand what are possible variations. They want to model differences. They want to understand differences. What are the differences between brains of people low fitness index and brains of people with high fitness index? What are the differences between malignant cancer cells versus benign cancer cells, right? You want to model relationship, right? How does your brain typically change as you age? And some things are known, but maybe more things can be discovered if you use a proper mathematical framework. And then, just like, sometimes you solve, like you have a signal processing problem, and you say, okay, to solve it in initial domain, in signal domain, may be very hard, like a convolution problem. But if I solve it in Fourier domain, Fourier transform domain, its solution is very easy. And then once I solve it in Fourier domain, I can actually invert that solution and see it back in my signal domain. Well, because we have a transform here, we can, whatever regression we do in the space, we can invert it back and do the same, right? So this is the big picture that I want to emphasize here. You have your data, and these are just, these blobs here are your data classes. Each point here would be an image, right? Or a signal, or an image in our case here that we're talking about. You take this transform, which is kind of a tangent type expansion for the data where things are linearized, and you do whatever you want here. In this case, I'm demonstrating that you can fit a linear classifier to this data. But then if you want to see what this linear classifier means back in image space, because we have this inverse transformation, we just invert the classifier. We can plot that as well, and I'll show you examples. But this is the big picture. This is what we call transport-based morphometry, and this is what we have been applied to it. First 1D signals, 2D images, 3D images, and so on, right? So it's this is a nice summary for what we're doing. Let's look at an example, right? So nowadays, Metin probably is familiar with this, like we have, most medicine departments will have a pathology and imaging core for a pathology department, where you have a whole slide scanner, right? Where you submit a scanner submit a slide to the scanner, and out comes a huge image, like 100,000 pixels by 100,000 pixels, something like that. And if you zoom in this image, there's a lot of information, a lot of detail, right? And in fact, you will see millions or at least 100,000 or so nuclei, and you can go and extract all these nuclei with some automated segmentation of them. And you have many patients, many tissues, many slides, and you can create these enormous databases of highly detailed information, right? The images here are plotted slow, but you can have easily like 200 pixels by 200 pixels, right? So there's a lot of chromatin information present here. How do we mine it? What do we do? Well, one thing we can do is just do the black box type machine learning approach. We're going to do this transport-based morphometry here. So first thing is, you can look at the data this way, but it doesn't give you a very methodical, very structured, very quantitative approach to understanding what the variations are here. How about you just take the data and do PCA, sort of like in the Eigenface's famous paper? Well, we're not going to do PCA on the image itself because in some ways it doesn't mean much, but we're going to do PCA on our transform. In other words, in the deformation space to the reference. Now you can start organizing this data in some main modes of variation. The modes are in this axis, right? So there are many modes. The primary mode of variation that explains most of the variance is this displacement of intensity, or in this case, mass, right? I should always use mass from the interior of the nucleus, so small nucleus, to large nucleus, right? This is what PCA in transform space recovers. The second mode is something related to elongation, right? So these are pretty round. You can get elongation horizontally or vertically. Third mode is some kind of placement of chromatin from the southwest corner to the northwest corner, right? Fourth one is placement of chromatin from middle to edge. Some perturbation to the left, perturbation to the right. There are many, many of these. How many? Infinitely many in theory, right? But we applied PCA to a few thousand of these things and came up with this. So I'm not showing the picture here, but most of us can understand that if I had applied PCA on this native image space, I'm going to just get a bunch of blurry stuff. You're not getting blurry stuff. You're getting the modes of mass pushing around, right? So the geodesics here, the way, if you model through this way, you get much cleaner picture and understanding. So this is something you can do. So these are your learned fixtures. And these images here are reconstructed by this inverse of this, these inverses of these maps, right? Here's your reference, you apply the inverse, and so first you do PCA on the features, then use the mean plus some direction, right? This is direction 1, this is direction 2, and so on. And how far you go along this direction is alpha. So you build your transport map, invert it, and apply it to your reference, and this is how you plot these images. If instead of modeling variations, you want to model discriminating information, this can also be used in a nice way. For example, let's say go back to our example of analyzing the differences or trying to understand the differences more precisely between benign cancer cells and malignant cancer cells, right? Again, you look here by eye, it's very hard to come up with some rules. You don't know, like any, I think, reasonable, humble person would say, well, first thing is, I don't know, I may get a gist or this or that. But so let's apply this pipeline, this TBM pipeline. Take your data, transform it, right? Put the transform space. What is transform space? These are these axes, right? Which again, you can invert and see. So one axis could be this kind of protein rearrangement or mass rearrangement. The other is X. How many? In theory, these are infinite functions, infinitely many, right? But how many we'll get is the same dimensions of pixels here. And then in this space, each point here corresponds to one of these images, right? And again, I can go from this space to the points here and back. There is no loss of information, just like you can go from an image to a Fourier transformed image and back. There is no loss of information. But in this space now, I can do my pattern recognition, and I'm just going to fit a linear classified, for example, this green line. The beauty about, and if you project the data onto this green line, you're going to get a histogram that looks like this. And so far, I haven't told you anything new or exciting. But the beauty about this, since this is a transform, I can actually invert the classifier, meaning I can sample this line here. What image corresponds to this point where my mouse is now? What image corresponds to this point where my mouse is? Well, these images are shown here, right? So now you can visualize the actual classifier that you apply to your data, and you can infer meaning from it. a pathologist might look at this and say, "Whoa, this makes a lot of sense." Class 2 here is the malignant version. Class 1 is the benign version, I believe. I forget exactly, but I think that's correct. So this is meaning it's saying that on average, the malignant version you're going to observe a phenotype that looks like this, and on average, the benign version you're going to look at a phenotype that looks like this. And a pathologist might say, "Well, this makes a lot of sense because cancer is a process characterized by fast division of cells, right? So when you take a picture, a snapshot in time, which is what this digital pathology must provide you, in a malignant tissue, you're going to be more likely to catch cells in the phase of division, in the state closer to the red state. Why? Because they divide faster. Out of 100,000 cells, you're going to catch more of them dividing or close to division. And it turns out that cells close to division, we're talking about a group today, the first thing that needs to happen is the chromosomes need to be spread, the chromatin needs to be spread, and microtubules have to go grab the right chromosomes and pull them apart. That's consistent with this phenotype, right? This phenotype here is more consistent with cells in other phases, not in the division phase. So now you can confirm with the classifier that is it recovering reasonable stuff, and in this case, it's recovering. And again, the equation, so now this W here is the classifier, not your PCA component, but the equation to reinvert and reconstruct any of these images are here. Any point in this transform space, you can revert, right? So you want to invert a line, no problem, invert it. So I showed you two examples, one modeling variations, the other one is modeling discriminating information. The third example is I want to model covariations. Right, so this is an example long ago by a collaborator from Stanford that actually gave us some data to test out these ideas. And here the experiment is imaging both myosin and actin in neutrophils. And these are cultured cells, but they are the process of moving. Of course, you take a snapshot of images, that's now you fix time, but you take many of them. And the question here was, okay, I'm imaging two channels, myosin and actin. Here we're overlaying them, right? But I can separate them. These are two channels. Here's the actin channel, the myosin channel. And the question here is we want to know is that a function that relates them? In other words, if I give you the actin channel, can I predict the myosin or vice versa? Are they related or are they completely unrelated? Right? So we can use transform-based morphometry to do this job as well. So we take the images, we take the transform. So these are the transforms. Remember the push forwards to the reference. And then in this transform space, we can do a very simple statistical analysis technique, which most of you will know is canonical correlation analysis. What is canonical correlation analysis? We find the subspace in this transform space that are most correlated with each other. And these turn out to be this blue line here and blue line here, right? So I go here, project the data. What's the correlation with that? We find the maximal correlated subspaces. It's like a PCA technique again, but for that job, find the max correlation. Once we find these lines, we then apply the inverse transform and see what they mean. And in this case, it gives us a very reasonable answer or a discovery for some plausible answer. It says that, in fact, we can, and this is held out data. This is not data using for actually performing the CCA, but we held some data out and then projected back into the space to compute the correlation. We obtain these functions, and these functions, the inversion of these functions again are shown down here. You can see that axis, right? So, and the inversion of the other channel myosin is here. And it says that to a very good degree, if I obtain this image here, I can predict what the image will be for this. And this prediction is actually very easy to visualize if I superimpose these two channels, right? It's giving some kind of antipodal relationship, meaning when actin goes to this side, as the cell is moving, maybe going that direction, myosin goes to the back of the bus, right? So that's an antipodal relationship that was discovered completely hands-free from this TBM approach. Another one that we did a calculation a long time ago is understanding the effect of drugs in cells, right? So here, cultured drugs, and where we apply the drug from 1 nanomolar, 4 nanomolar, 5, 15, 62 to 50, and so on. So you can see that something's happening to these cells, right? In fact, we know this particular experiment is this Wortmannin drug it's an antagonist that prevents a certain protein, this forkhead protein, from translating throughout the cell, right? So if it's in the nucleus, it will stay in the nucleus. when you apply this drug. So that effect is known, and this is the picture on the side of the box of the drug, right? So our question is, could we have discovered this effect with TBM just hands off like we did here? And the answer is yes. So we segmented the cells, right? And here you can see more close up of the data, right? This is 0 nanomolar, no drug applied, 1 nanomolar, so on. So what did we do? We did the pipeline. Take the data, do transform. In transform, find the regression that most correlates with the molarity of the drug we applied. Invert that regression, right? And here is that regression, the inversion rates. And this red here is the average nucleus superimposed. So it's telling you that when you do apply the drug, we split into two classes, high application and low application. When you do apply the drug, the images look like more like this. When you don't apply the drug, the image is more like this. Here you see the protein all over the cell. Here you only see the protein in the nucleus, right? And you can see a more detailed picture of how these distributions look like here. And in fact, if you want to show that particular regression, right, you see here in the x-axis, you have the molarity of the drug, and here's the reconstruction of what, on average, the average cell will look like, completely hands-free. And this is, in fact, this is the data provided by the company as far as they have a percent activity, but their percent activity was people looking at it and quantifying by eye or some gestalt feeling about how much protein was not allowed to translate. Here, this is an actual picture with actual units to them, right? But the fitted sigmoid that we get is very, very close to the real data. I'm not going to have time to go much into this. I'm just going to point to a recent paper. You can apply this to do very, very interesting things in cancer, right? One paper we recently wrote is, okay, we can take data from a variety of cancers and a variety of institutions and a variety of databases, right? So, but here we focus on 4 different kinds of cancers. And now this transport-based morphometry also gives you this ability to look at a very specific question and combine data from different, without the need to data harmonization or transfer learning or anything like that. You're looking at something almost like physical. It's almost like a physical quantity with physical units, right? So as long as you have mass distribution images, you can compare that to any other data set. So we're able to do this pan-cancer studies and figure out what does malignant liver have to do with thyroid data? Are they the same kind of nuclear processes that are involved? So this approach is allowing for this new kind of studies to be performed. And if you're interested in that, I can give you a specific talk on that. But there's a new paper coming out, and this is for prognosis, diagnosis. You can study, do pan-cancer studies, study similarities between cancers, differences between cancers. It opens up a Pandora's box for many types of studies. I'm not going to go much into that. I'm just going to go through Bayes morphology really quick and then wrap it up, because otherwise I may run out of time. Here is a recent study from one of our past lab members. She's now a professor at Washington University in St. Louis, where she was looking at imaging copy number variations in genetic mediated autism. What was the question? She wanted to look at gray and white matter morphology and see if there's a particular morphology associated with autism when autism is due to deletions of genes or versus duplications, right? So we have duplications, deletions, and we had controls as well. And we want to see, does a deletion type perturbation cause a particular signature in brain morphology, right? So it's the same thing, but now we're going to do with brains in 3D and so on. But these images, here you have gray matter distribution. The word for the image is gray matter distribution, gray mass distributions, right? I'm trying to be pedantic here to drive the point in that almost all these imaging modalities, you are looking by definition at mass distributions, with the understanding that you can't integrate this mass and come up with a kilogram measurement, right? The image is not calibrated. But besides that, you're looking at mass distributions in cells, in tissues, organs, and so on. These are images of mass. So it's perfectly natural to apply this approach. So here's a little bit about the data, right? And the distribution of other other clinical variables so that you have a sense of the data set, right? See, this is a collaboration with UCSF. There were 206 individuals, 48 deletions, 40 duplication individuals, and 818 controls, right? And it's known that these kind of deletions and duplications are strongly associated with autism and other conditions. You know, and one hypothesis that we're throwing here is that perhaps that signature, whether it's a deletion or duplication, can actually be seen from, in this case, gray matter morphology. We can do any kind of imaging, but we're doing gray matter morphology. You know, if you just look at simple features like brain volume, right, you can get some kind of differences, but you know, you wouldn't expect that. In fact, you can't really differentiate controls from this, and you can get a signal to differentiate deletions from duplications, but very, very weak. The distributions overlap by quite a lot. So we did the transport-based morphometry approach, which is, you know, it doesn't differ. If some of you know the deformation-based morphology, it doesn't differ that much. There's one crucial piece that is different. Instead of deformation, we're talking about transportations, right? Instead of registration, we're talking about transportations. But otherwise, it's a similar pipeline. You take your T1 weighted images, you do your gray and white matter segmentation, then you take your gray matter and you map it to a reference, in this case, an atlas of the brain. And this is your embedding for you have dx, dy, dz, right? So 3 of these embeddings, and then you do your business here. You do your statistical regression to see what you want to see. So first business is, if you're trying to do PCA on all this data, you can do PCA in image space, or you can do PCA in transform space. And if you ask, like, what does one component reconstruct in percentage of the original variance in the data set? In the image domain, you get 10%. In the transport domain with one component, you get 45%, close to 50%, right? So you can see that these transport modes, they have a lot more closer connection with the actual data because the fewer of them reconstruct the data to a higher precision of accuracy. So here we're going to do supervised learning, right? But we're going to do supervised learning in transform space. And first thing we're going to do is do this decision boundaries, right? So I'm going to plot the PCA type decomposition and try to solve this, right? And one of the approaches that we came up with is so-called penalized discriminant analysis. I'm not going to go into that. And then in this PLDA space, perform a near centroid classification, right? So if we find a point that lands here, well, we measure this near centroid computation. This is the boundaries implied by the near centroid, and we assign to the near centroid. You know, and the basic take-home message is we could get a very decent classification accuracy in this 3-class problem using just gray matter morphology through the TBM approach, right? And so this is success in some shallow meaning of the word. But if this wasn't transport-based morphometry, I would stop the talk here and ask questions, right? And one natural question that a person would ask, he says, okay, what is your deep learning approach? If this was done with deep learning, imagine, what does it tell you about what this thing, what is it saying? What is the difference, essentially, right? You found some difference. You can defend it statistically speaking with held out data, right? Non-used for training. Maybe 94% is this inaccuracy. You can say, okay, so it's interesting, but what is the difference? Well, typically machine learning doesn't give you this opportunity, but now this does, because we can invert these classifiers, right? So this is the discriminant direction that you're looking at. Of course, now you have 3D data, right? So, but this is across the discriminant direction. And you can see the shifts in mass distributions in specific areas, right? So you can see increased spleen density in certain areas of the brain. That deletions implies increased spleen density in the brain as opposed to duplications, right? The controls are in the middle. So now you can study and see, you know, some density, you know, in the deletions or in some thinning and so on in the duplications, right? This gives you a lot of a lot of opportunity to see what the differences are in mass, gray mass distributions, right? So you can look and come up with hypotheses. Potentially, this hypothesis could point to foci, to locations where to direct treatment and so on, right? So now you can have some understanding of this. I'm not going to go into this, but you can apply this to any density image, right? This is another study we published a few years ago, trying to predict, so let's say we image subjects. These are knee cartilages. We took the cartilage, flattened them out, and we wanted to say, hey, which ones will progress to have osteoarthritis symptoms 3 years from now? Can we make this call now? And in fact, we can do the similar kind of predictive modeling. We can also, turn these pictures into understandable, what is the T2 density? Where should you look for spot problem areas, and how does that predict? So that's the approach that we're defending here. Big picture summary, right? So a lot of us, I'm not saying this is all of image data science, but a large portion of it is taking databases and asking questions from it. The questions typically are, you have 2 classes, we have 10, right? But start with 2. You can ask questions like, is there a difference between persons with high fitness index? Is there a difference with autism, persons that have duplications versus deletions, right? Is there a difference between nuclear chromatin distribution in benign cells versus? That's the beginning, right? You ask a good question, you have a chance for a good answer. This typical, and if you do find a difference, if you do find some high classification difference, accuracy that points to a difference, then you ask the question, what is the difference? Please teach me what that difference is. This is what we try to address with this kind of transport-based morphometry. I make the argument that it's a good idea because we're using methodology that is closely related to the underlying physical processes that are giving you the data that you're measuring. What's happening in brains? Transport. What's happening in cells? Transport. The reason why one brain looks different from another brain is because there has been some differences in transport. Of course, there's also been differences in sources and sink, but we can't access that. But at least the transport part we can access and try to mine the differences, right? So the reason, I'm not showing you all the comparisons here to traditional machine learning because this does much, much better, right? In terms of accuracies, we can throw any kind of deep learning to any of these problems, the accuracy will be a lot lower. My intuition says the reason is because we're starting, it's almost like cheating. We're starting from the right physical process, right? The other one has to learn what the physical process is. We know what it is, at least in part. So you're in terms of accuracy, you're going to do a lot better. The summary of the gist of the idea is you take, it's the same as the deformation-based morphometry. Take an atlas, do the transportation to this atlas, and this is your transform. But it's a transform that actually reconstructs the entire data because you can push the atlas back to the exact image. You can't do that with deformation-based morphometry. Here you can with transport. So you do have like the space of input functions and output transform, and you have an inverse. And that's the basis for this whole thing, right? We have more mathematics for what we're saying here. This is not math talk, but there we can defend. There are a lot of theorems to show when classification will work, when it will fail, etc. Right? Which is also something you don't have with AI type approaches. And here's a summary of TBM, right? You take your input data, you can do exploratory analysis, PCA, subspace learning, modeling texture and shape naturally, simultaneously. You can visualize variations in the data set. You can see, oh, there's big, small, there's elongated, there's shift to chromatin middle to fat, blah, blah, blah. If it's kind of looking like spherical harmonics. You can do discriminating analysis. You can say, okay, my question is pertaining to, I want to discover what the differences are. Well, go fit a classifier and invert it and visualize it. Now you can see what the differences are. Not only have a predictive model to say this patient has this kind of cancer, but you also can say it has it because it looks like this. You can also do functional relationships, right? CCA, map things over time, drug molarity, and so on. I gave you many of these examples. Last slide is, we do have some software, that we are providing. It's called PyTranskit for that does some of these TBM approaches, does these transforms, and so you can, just call a transform, just like you call a FFT, right? You can call a CDT. Right, like we have for 1D signals, 2D images, 3D images, and so on as part of this. And we're building this TBM toolbox that hopefully some will have an interface that one day you can see, load your data, start, fit the classifier, invert the classifier, and so on through clicking, right? So hopefully this will come up very closely. Last slide is thank you for your attention. Thank you for Matt for inviting me as well.
I'm clapping on behalf of everybody. This was such an exciting presentation, and some other people are also clapping. TBM is a really exciting framework, and I think several of us will be looking at it closely because just it's incredibly well, it's a very sound theoretical framework, and it's just it is not we are throwing a bunch of numbers and let's see what it will say type of thing. So that is super exciting. And then the functional part, the prediction of the G, just when I was thinking about it, just kind of I was thinking about Kolmogorov-Arnold networks. I don't know if you are familiar with those.
I've heard about them. I'm not 100% sure what they mean yet, but I've heard about them.
Yeah, instead of learnable parameters, they are saying, can we learn the activation functions in CNN, for instance?
Right.
So when I was listening to your talk, and it's just my mind was going there. It's a different framework, but I think there is something just I think you found a really interesting point here that let's look at these functions and let's try to learn these functions and then go from there. So I think that is a incredibly powerful framework. Thank you for sharing this. I think it's going to inspire several people to look at your TBM. It's good that you also have the software available. So I think if people are interested, they have no excuse not to use it. And if you use it, please cite his papers. So let's see if people have any questions. I certainly do have some questions, but before we do that, Mustafa, are you in a place that you can turn your microphone on, ask your question?
Yes, hi. Thank you very much for your presentation. Honestly, that was a very great and exciting presentation. I'm planning to go through your papers. It seems very interesting. So my only question is regarding like this transport or this transformation. So because you're switching between like image space and this transformation space, my question is if always these transformations are going to be invertible or not. So because I'm assuming that is kind of like in terms of like matrix language, I'm assuming that we are talking about like a matrix, for example, as a transformation. So I wonder if that is going to be always invertible to just switch between like image space and latent space or that transformation space.
Yeah, well, first of all, thank you for the compliment. I appreciate it. And the answer is, the first answer is yes. We have mathematics to say some things, and let me say that thing very clearly. In the theory part, if we ask the question, if you give me a function, does there exist for any function that you give me? And in this case, we're looking at functions that are mass densities, right? Because this is what we're imaging. But if you give me a mass density, can I come up with a unique diffeomorphisms that pushes whatever reference I use to that mass density? And under very reasonable assumptions, the answer is yes. Okay. If you give me a density that is smooth, that that has a confined space, that has finite energy, and so on. Meaning if you, of course, if you give me a function that has some infinite and some bounded and so on, then the answer is no. But under some very reasonable function, if you give me a reasonable function, an image like this,
okay,
right, then the answer is there's always a unique f that pushes the reference that I chose onto that function, which means there's always, for one function, again, a reasonable function that you gave me, there's always a unique point in the diffeomorphism space that is uniquely associated with that. So there's also an inverse.
That is
So
always like one-to-one function, right?
one-to-one,
Always?
always. Just the same, like for example, when you have a Fourier transform, you give a function. Is there always a function in Fourier transform space that corresponds to that input? The answer is under reasonable assumptions. Of course, if the function is not integrable, no go, right? So there's some reasonable assumptions there too. Same thing here. There are some reasonable functions. The domain have to be bounded. The function has to be finite, etc. You know, you cannot have.
Okay.
And in this case, we're talking about continuous functions, or they can have discontinuities, but not everywhere, right? Almost everywhere. So under some very reasonable assumptions, you give me a function, I'll give you a unique point in transform space. Yes.
Hmm. Thank you very much. I will read this paper definitely. Thank you.
Yeah, and to clarify, like this is not like, for example, sometimes people will do this connectivity graph approach transforms, where you say, you know, I'm going to give you a manifold learning kind of approach, where you say, I'm going to give you a latent space that I recover some manifold learning. And that is also a mapping from one space to another space. This is not it, because we don't need data to learn anything. This is just, you think of it the same way as the Fourier transform. The Fourier transform, you don't need data to compute it, right? It's a mathematics thing. Here's the same thing. You don't need data to compute. Of course, you need to compute the data. You need one image, but you don't need other images to compute the transform of this image, right? So it's a mathematics thing. So in that sense, there's always a functioning transform space. But those are the papers. I didn't talk about this representation. There's, we wrote several papers talking about the representation, like defining this transform, showing the mathematical properties. This talk was just essentially an application of that. Say, okay, we can do TBM. Once you can represent things like this, here it provides you the opportunity to do TBM, right? But I think maybe to interpolate on your question a little bit, to say, okay, if you go to this transform space here, right?
Mm-hmm. Yes.
Are there points in the space here which you can't invert and get an image back? Yes, right? Mm-hmm. Because so you have to have diffeomorphisms here. So if you're going to do something here that fits a classifier and gives you a function that lands up way out here, then you can't invert that, right? So that you may be asking that as well, right? So here, any function, any point in the space which gives you an f, which is one-to-one, no problem, you can invert that. But there are some points in the space here where the f is not one-to-one, and those you can't invert.
Yes.
But that will come about if you use the wrong modeling technique there. If you use PCA and go too far, for example, right? That could happen.
Yeah, thank you very much. I really like the way that you use kind of like a line search method here, like this alpha WX is kind of like optimization method line search. So that reminds me of kind of like diffusion model with interpretable direction. I'm not sure if you have heard about that, but diffusion models are coming from physics, and this concept diffusion models with interpretable directions, that is kind of like a little like similar to this research that you are doing. Thank you very much.
Thank you for the question.
Unfortunately, we are out of time. But just, we have, I have other questions, and the other people have other questions. For those of you who missed the first slides where Dr. Rohde put his email, so you can reach out to us. Are you going to be able to put back to that slide? Yes. So it's just his first name and period last name at gmail.com. So I presume they can email you.
Yeah, for sure. Please do. We can set up a separate Zoom call. I'm happy to talk to people.
Thank you. And then this is a really exciting framework, and I will certainly be looking into. And then let's just put your email in the chat as well. So I will be looking at the TBM for many of the problems. It's a fantastic framework. Congratulations, Rohde, and thank you so much again for joining us this afternoon and giving a really wonderful presentation. We really appreciate it. Thank you. Thank you very much, everybody. We will meet in our next meeting soon. Take good care.